Throughout our development, tissue morphogenesis plays an important role. These tissue movements are ultimately brought about by changes in the shape and mechanical properties at the cellular level, which in turn depend on cytoskeletal elements for force generation. One key morphogenetic process is the cell division, where one cell divides into two. It is of extreme necessity that cell division occurs with utmost precision to sustain normal tissue development and homeostasis, as errors in the same can ultimately lead to development of lethal diseases such as cancer. Studies conducted on the archaeon Sulphobolus were amongst the first, indicating remarkable similarities between the cell division machinery of archaea and eukaryotes. Sulphobolus, being a thermophile, requires high temperatures to thrive in, and thus studying the process of cell division in them using live cell imaging had been impossible until recently. A model showing ESCRT-III mediated cell division in S. acidocaldarius had been recently published. Also, CdvB1 and CdvB2 have been assumed to have identical membrane scission abilities since they are close paralogs sharing 65% amino acid identity and 80% similarity, which is why the Sulphoscope had been especially constructed for live cell imaging of Sulphobolus to test the individual functions of CdvB1 and CdvB2 in cytokinesis using individual in-frame deletion mutant strains. CdvB1 deletion strains maintained symmetric division but showed occasional division failures wherein cytokinesis halted midway and the nascent daughter cells fused back together to give a single cell carrying two spatially separated and compact chromosomes. On the other hand, CdvB2 deletion strains had severely impaired growth. These showed asymmetric division with high variance in the daughter cell size. Thus, they concluded that CdvB1 must be important for force generation during cytokinesis, and CdvB2 must be important for fixing the contractile division ring in place. This finding is also remarkably parallel to the mammalian ESCRT-III proteins CHMP2A and CHMP2B recently found to have different functions in membrane remodeling. Thus, Sulphobolus can be used as a simple and well-defined system to study ESCRT-III dependent division and the core eukaryotic division machinery functions.